brought to you by Curious Maryland. Now, take a look at this picture. This is actually a model, and this is a cell membrane. So before we go any further, let's label the parts. And then we're going to go ahead and talk about a couple things on this PowerPoint. One would be the structure of the cell membrane, but I actually want to really focus on how do you get this phospholipid? And I'm going to use some models for that. So that's the picture that we're just labeling. So let's do this. Go back to the camera and label the two most important parts. So here we go. Okay. And you might notice I have a part there and a part there. It's a lipid bilayer. Here's one layer. Here's the second layer. So I need to put that there. Label that. So what I want to do next is go to the models. I'm going to pause and switch around and show you how we get to the actual structure of one of these with the models. So let me just kind of show some other things. So that's what we just labeled. Here they are lined up. All right. You actually have two parts, the head, which is a phosphate group and a polar or charged molecule, and it's hydrophilic, it's water-liking. Then you have the tail that is hydrophobic, and it doesn't like water. So let's do this. I'm going to reposition, and we're going to come right back. Now this is a lipid. This is a 1, 2, 3. This is a triglyceride. Now how do you get from here to that phospholipid? See, you're going to have to take one of these off and attach this. So this looks kind of complicated. Let me go ahead and go back to the PowerPoint. If you recall, a triglyceride is a glycerol with a fatty acid chain attached to it. All right? So now what we're going to do is we're going to actually remove one of those and transform it. But let's take a look at this first. See this phosphate? So this is glycerol phosphate. So imagine, so each one of these has a fatty acid chain. So this one right here, we take the fatty acid off and we add phosphate. But we're going to add something else. So here, this is what I'm trying to build. All right? And let's actually go to here and actually look at the actual structure. Here is glycerol. Fatty acid chain, that's the hydrophilic part. Hydrophobic, that's the hydrophobic part. And then what makes the hydrophilic head of a phospholipid is this phosphate and then this thing called choline. So let's actually go to the models and see if we can do this. All right, so I'm going to dangle it down like this. And so here's carbon 1, 2, 3. So I'm going to, carbon 1, 2, and 3. So what I got to do is take this off. Okay, got rid of one of the fatty acids, and now here's phosphate, and then this is choline. So let's attach it like this. Okay, now let's try and position it a little better here. All right, so here's your tails. This is the hydrophobic tail, and this is the hydrophilic head of a lipid. All right, that's our model. So if you remember, this would be pointing down if it was this part that I just took off. Okay, so here's there you go. So I'm going to pretty much finish this up. Let's go back to the PowerPoint. That's what we just made. We went from this to a phospholipid. And what is the importance of that? Here's a really good picture. I've got a link here that you can look at. So you got this is the glycerol, and then you have the fatty acid chain, and you've added in the phosphate. So let's do this. The importance of this is your cell membrane is selectively permeable. It's fluid. So pretty much going to end with this, and I'll talk more about why is this important, because this allows certain things in and out. This is passive and active transport, and so I'll do another video on that. So that's pretty much a short video on the structure of a phospholipid and how you get a phospholipid from a standard lipid to add in. Here's this is the phosphate, and there you go. Now we have a phospholipid. We have one of several things that combine to make it a selectively permeable cell membrane.